Yep. Hello, everybody. My name is Ekta. I'm here representing the Boolean Lab from UC San Diego. Today, I'll be talking about my project on an AI-assisted investigation of tumor-associated macrophages and their polarization in the context of colorectal cancer. This research was conducted by yours truly and Dr. Tirtharaj Dash under the primary advisory of Dr. Debashish Sahu. And in this presentation, I'll briefly discuss the context and need, the methodology used, and the primary results from this exciting study. Colorectal cancer, or which I'll be abbreviating as CRC, is the third most common cancer worldwide, comprising over 10% of incidences back in 2020. It involves genetic alterations in cancer cells and also in the cells living in the surrounding tumor microenvironment, such as stromal cells and immune cells. Macrophages in specific, particularly tumor-associated macrophages, or TAMs, play a key role in colorectal cancer onset. TAMs are derived from blood monocytes, and they are recruited to the tumor site, where they are responsible for promoting tumor proliferation, migration, and invasion. They can adopt a wide range of different functions based on the environmental stimuli and can be categorized as M0, M1, and M2 macrophages. M0 macrophages are unstimulated. M1 macrophages are immunoreactive and have pro-inflammatory characteristics, and M2 macrophages are immunotolerant and have anti-inflammatory characteristics. This literature-supported dynamic of macrophage polarization inspired researchers in the Boolean Lab and the Ghosh Lab at UCSD to develop a computational model that identifies genes that are related to this polarization dynamic. They found that the traditional nomenclature of immunoreactivity and immunotolerance oversimplified the complex behaviors of macrophages. Thus, they basically created a model based on Boolean implication analysis called the Signature of Macrophage Reactivity and Tolerance, or the SMART model. This model was trained on human macrophage samples, and it identified clusters of genes that optimally captures the continuum of polarization states. The researchers then recognized 48 genes that are associated with the immunoreactive phenotype and 290 genes that are associated with the immunotolerant phenotype. In this mapped model shown on this slide, cluster 13 represents the immunoreactive genes, while clusters 14 and 3 represent the immunotolerant genes. So the SMART 13-14-3 signature is an extremely valuable tool for characterizing macrophage polarization dynamics across many different types of tissues, organs, species, and immune cells. Therefore, we can assume it to be a universal gene signature. While it has been validated across many different data sets, the signature's utility is limited in data lacking macrophage specificity, such as large cohort disease data. This limitation motivated my research to create a specialized signature for TAMs specifically in colorectal cancer. On this slide, I am highlighting the fundamental steps that were used in my project. We applied the SMART model to computationally extracted macrophage cells from single-cell RNA sequencing datasets with both cancerous and human colon samples. My findings at the single-cell level basically translated into refining the universal 338 gene signature into a specialized TAM signature that optimally characterizes polarization in this disease. So in addition to the foundational SMART model that was built in-house, my project is also supported by other pre-existing findings. So all data sets were collected from the NCBI GEO database. The gene expression matrices used for these projects were extracted from single-cell RNA-seq, bulk RNA-seq, and microarray colorectal cancer studies. To focus specifically on TAMs, we artificially filtered macrophages from single-cell datasets. This was based on the work of Dr. Dung from the Boolean Lab. Dr. Dung identified ThyroBP and FCR1G as universal macrophage gene biomarkers. After implementing a threshold for the normalized gene expression for these particular models, we applied the SMART model to extract TAMs. And furthermore, our refinement pipeline was already also heavily relied on t-test dependent differential gene expression analysis. So I'll be discussing these steps in the future slides. So to quantify the signature's predictive potential in, again, capturing the polarization dynamics of TAMs, we generated a composite score for the cluster 13 and cluster 14-3 genes in the SMART model. Again, cluster 13 genes are associated with immunoreactivity, and the genes in clusters 14 and 3 are associated with immunotolerance. This composite score essentially involved summing up the normalized expression values in the specific gene clusters and using weights to differentiate between the two polarization states. A negative weight of -1 is assigned to the immunoreactive genes, while a positive weight of 1 and 2 are used for the immunotolerant gene clusters. A more negative weight or more negative composite score indicates a higher immunoreactivity, and a more positive score indicates higher immunotolerance. We determine whether a sample has high or low composite scores by defining a threshold. This threshold is assigned using a computational tool called the StepMiner tool, which was built by Dr. Sahu, and essentially it uses a complex regression approach to detect distinct transitions in the scores in a particular data set. So plotting the two composite scores and the StepMiner thresholds against each other allows us to visualize the polarization dynamics in both healthy and tumorous macrophages at the single cell level. So here I am showing those results where the x-axis represents the immunoreactive composite score and the y-axis represents the immunotolerant composite score. The green dots represent healthy colon TAMs and the golden dots represent colorectal cancer TAMs. As a reminder, a more negative score is associated with immunoreactivity and a more positive score is associated with immunotolerance. So using the assigned StepMiner threshold that we applied, we can identify the bottom left quadrant on this plot as being highly immunoreactive macrophages. And again, this is because these cells have a negative cluster 13 score and a more negative cluster 14-3 score. And using the same logic, we can apply this top right corner as being highly tolerant macrophages. This specification unveiled a really interesting finding, and it actually allowed us to link polarization to cancer prognosis. And what we see is that tumorous macrophages have a higher immunoreactivity than those from the healthy colon. So on the right, we show that this pattern is consistent in both human and mouse data sets, and this inspired our notion for refining our gene signature. So now you may be wondering, if the SMART model can provide this sort of information, what purpose would a refined signature serve? Well, previously, we were looking at extracted macrophage cells. So again, we took our single cell data, we used thresholds for ThyroBP and FCR1G and applied the SMART model to macrophage-specific cells. This is a biased perspective and usage of the SMART model, whose purpose is to measure polarization for macrophages. We observed the constraints of the universal signature when particularly looking at large RNA-seq data sets. In these data sets, we are not able to extract macrophages. And what we find is that the SMART signature cannot differentiate healthy colon and colorectal cancer samples using this notion of composite scoring. This limitation is illustrated on this violin plot here, where green represents the normal colon samples, the top bar, violent circle, and the bottom golden represents the colorectal cancer samples, and the purple dots represent the total composite score of the SMART signature, which is a summation of the immunoreactive and the immunotolerant scores. A lack of separation between the healthy and colorectal cancers, which is highlighted in this bar here, is also quantified with the area under the ROC curve, the AUC metric, where a higher AUC indicates better predictive power and classification. So a AUC of 0.66 is, we don't consider that as statistically significant. So to address this limitation of the SMART model, we turn to removing genes whose expression may not be responsible for the polarization in colorectal cancer. We conducted differential gene expression analysis on a selected training set which is enriched with highly immunoreactive and immunotolerant macrophage cells. From a set of stringent filters, including log fold change and its statistical significance, as shown in these volcano plots, we composed a 24 gene signature of the total 338 genes from the SMART signature. To further demonstrate the need for refinement, we also created a noisy 31 gene signature. And what we found is that the SMART signature and the noisy signature failed to capture the expected polarization dynamics that were observed at the single cell level. In fact, they show the reverse pattern where we actually see that normal colon samples are technically more immunoreactive. What we expect to see is a skewness of cancerous samples towards the left or higher immunoreactivity and healthy colon samples to the right. Our refined signature successfully restores this expected dynamic and validates our findings at the single cell level, shown on this plot right here. So as a reminder, I previously showed how the SMART signature is incapable of discriminating the healthy and cancerous phenotypes in bulk, large RNA-seq data sets. This is the same plot that I showed before. With our refined signature, we are now able to. And we see that the tumor colon samples have a more negative composite score compared to the healthy colon samples, which means that these cells, which are likely to be TAMs, are more immunoreactive and carry pro-inflammatory characteristics. So our refined gene signature is showing the dynamics that we saw at the single cell level, and this is validated across many other RNA-seq data sets as well. So while our study primarily focused on linking cancer prognosis to macrophage polarization, we discovered that our TAM signature can also classify colorectal cancer subtypes. For example, the disease develops through pathways like chromosomal instability, mismatch repair, and serrated pathways. Our refined signature distinguishes these pathways' effects on cancer development. So for example, the mismatch repair pathway has two states, stable and unstable. And the stable state, MSS, does not often trigger an immune response, while the unstable state, the MSI state, is associated with an unstable tumor. Our refined signature shows that the unstable colorectal cancer samples are more immunoreactive, which suggests better responses to immunotherapy. So while these findings are promising, further research is warranted to explore the unique features of colorectal cancer. But nonetheless, we are very confident in our signature's ability to predict prognosis and identify macrophage polarization dynamics in colon tumors. So yeah, thank you so much for your kind attention. I would also like to acknowledge my co-authors and mentors and the NIH for funding my research, and I'm happy to take any questions. Thanks.